I want to welcome everyone today to our virtual Lunch and Learn, working to bring back wild rice. We have a great group of panelists today, a great group of speakers. So I'm going to quickly just keep going for a couple of seconds. My name is Ellen Holste. I am the Community Program Manager at Pierce Cedar Creek Institute, and I'll basically be your moderator for today's session. So I'll help keep things going along, getting our panelists going, and helping get your questions hopefully answered today. All right, so working on bringing back wild rice. Our first speaker today is Alex Wheaton. Alex is the water resource specialist for the Gun Lake tribe. And I'm gonna stop sharing my screen so Alex can share his. Welcome, Alex. Thank you for joining us today.
So as she said, I'm Alex Wheaton. I am the water resources specialist with the Gun Lake tribe. I've been working for the tribe now for about 4 years. The word Monomin is the Pottawatomi word for wild rice. It translates into the good fruit or the good berry, as some of you may have heard. It also kind of means the food that grows on water. And it's a very important culturally species for the tribe. It is a key to the Anishinaabek migration story, how the tribes got where they are today. Thousands of years ago, the tribes lived over on the East Coast, and they were told to find the food that grows on water. And you can see in the top picture there, the tribes migrated westward and settled where they are today. The Gun Lake tribe settled here in the Gun Lake region as historically Gun Lake and the surrounding lakes were, before they were dammed and settled, full of wild rice. It also is very important culturally. It's a source of nutrition and sustenance. It's one of the things that's always present in ceremonies. Many citizens still harvest wild rice and rely on it for food throughout the year. There's 2 species, as far as we know right now. Other presenters will get to that in a little bit, but right now there's Zizania palustris and Zizania aquatica. Palustris is the northern wild rice. Sometimes you'll hear that referred to as lake rice. And then there's the southern wild rice, the aquatica, that is more apt to be found in rivers with flowing water. But obviously that can change depending on where you find it. We found that you find either of the species in both kind of habitats. The northern wild rice is usually a little bit smaller. The seeds really hold to the seed, to the stem. As you can see in the pictures, if you look closely in the very top, you'll see the seeds and then the flower below it. Whereas on the right, you can see the seeds kind of opening up above the flower. So that is kind of one way you can identify between the two. Also, the leaves on the aquatica are usually quite a bit wider than they are on palustris. But as you'll hear later when we start talking about genetics, there's kind of some blurry lines there that we're still trying to figure out. A little bit about the life cycle of wild rice. So wild rice is a species that germinates in mid to late April. You'll see the leaves shoot up on the bottom of the substrate underwater. The submerged leaf will keep growing until about mid-June when it starts to float, and you'll see a ribbon-like strand floating on the surface of the water. Late June, early July, depending on the year, you'll see an aerial shoot start to stick up out of the water. And once you hit this stage, it really moves along fast. And by late July, you'll start to see a flower sticking up, and usually about 45 days after you see the flower, the seed is ripe for harvest. There's multiple ecological benefits to wild rice. Obviously, it's a food for not only humans, but for waterfowl and other species. Even before the seed is present, multiple species will eat the plant, from muskrats to different waterfowl. And then once the seeds are there, you get your blackbirds, your ducks, humans harvest it, a little bit of everything. In addition to that, it's protection for wildlife. I was out all week harvesting wild rice, and there's obviously birds all over it. You see fish in there, it's using it for habitat. In the springtime, you see fish spawning in there. As well as its benefits to water quality. In river systems, you can see clearly when you're out in the beds that it's holding that substrate there, it's sucking up nutrients, etc. So there's a huge benefit to having wild rice. There's obviously lots of threats. As I mentioned earlier, historically, there was wild rice all over this region, and now it's pretty hard to find. Hydrologic changes. For example, in the bottom there, you see Gun Lake. The basin you see on the left historically was, 2 feet deep, the whole thing. They put a dam on Gun Lake, and now that's a little 2 to 4 feet, so it's a little at the higher edge of the depth that wild rice requires. Invasive species are a huge one, and that's both plant and animal. Mute swans can decimate a population in very little time. They eat the stalks before they even come up, and as soon as they come up, we've been to populations where we'll seed it and it'll be coming up like a green lawn above the water, and 2 mute swans will come in and it looks like a lawnmower has gone through and cut every stalk off. Shoreline development, again, if you look at the bottom picture of Gun Lake, it's almost 90% developed. So along with shoreline development comes things like boats. Obviously, if people are driving through a wild rice bed, it's not going to grow, it's going to get chopped down. Another big issue is climate change. With these warmer climates, we're seeing not only different insects showing up, but also fungal diseases that can really affect the germination rate as well as the pollination rate. So that's one thing we're trying to look into a little bit more. Again, wildlife activity, like I mentioned, muskrats, historically, they might have been trapped. As that kind of fades out, the popularity of trapping, we get more and more muskrats, and those can really decimate a population. And then we have problems like lack of knowledge. Many people don't know what wild rice is and can't identify it. And we see that causing a lot of problems. For one example, a big thing is invasive species control. If we have people going out and they're spraying things like Phragmites, wild rice can look very similar to Phragmites, and if they don't know proper identification, they might spray it on accident. Boaters don't know what it is, so they drive right through it. Fishermen cut it down to get access, so on and so forth. So the tribe has been working with wild rice for almost 10 years now. Currently, we get funding through the tribe as well as the Great Lakes Restoration Initiative. We have many different aspects of this project. We've been working with Pure Cedar Creek now for, I think this is 2 or 3 years on a couple of different projects, but to highlight a couple of the areas, genetics is a big one. We do some aerial imaging. I'll kind of get into a couple of these a little bit more. Also, restoration is probably the main part of ours. So transplanting, seeding, working with enclosures, invasive species control. So your common invasive species that compete for the same habitats, Phragmites, Typha, outreach and education to make sure people can ID wild rice, they know what it is, and it gives them kind of that want to protect it. And then we participate in what's called the Michigan Wild Rice Initiative, and that is a collaboration between all the tribes in Michigan, as well as nonprofits and state and federal government. And that group is working to develop kind of rules and regulations as far as wild rice goes. So protecting it at a local scale, and then creating regulations. In other states in the Midwest, like Minnesota and Wisconsin, there's actually regulations on how you can harvest wild rice, and there's none of that in Michigan. So right now it's kind of a free-for-all. You can go out and harvest as much as you want, whenever you want. In other states, there's pretty tight regulations on who can harvest, where they can harvest, when you can harvest, and what you can use to harvest. So I'm not gonna go into genetics a lot because we have 2 other panelists that are gonna talk about this a little bit more. We started doing this in 2019 because we started to see that the line that we thought was there between species might not be there. We were seeing different phenotypes pop up, and it was becoming really hard to identify between the two. And another thing we were seeing is that when we use seed for restoration, we get it from different places, and we try to do it locally. And we were starting to see that seed from different areas was doing better than seed we got from other areas. And we were trying to identify if there was some maybe genetic differentiation between those seed sources. And if we could pinpoint why that was happening, maybe we could utilize some of that data to help with our restoration. So we started doing that in 2018 or 2019, and we're kind of learning from our past research, and I'm hoping Charlene and Brad can talk about that a little bit more. Another thing we've been doing is these aerial assessments. And one of the reasons we started to do this was because there's a lot of areas, specifically in the Allegan State Game Area, that are really hard to get to. You can't really get to them by boat. It's almost impossible to walk to them. And we know there's rice in the area. So we figured if we could use drones to kind of create a model and be able to pick some of those different trying to think of the word here, different patterns through imaging, I guess I'll say, we would be able to fly those areas and be able to pick out rice. And we're starting to get to that. If you can see in the pictures here, it's really hard to see unless you're really close up. So the bottom picture is kind of a zoomed out picture, and then if you zoom in, it's really hard to see on the screen here. But if you really zoom in, it's kind of obvious to the trained eye where the rice is. We're also able to, in the larger stands that we fly year to year, we can monitor the size of the stand and the density. And also by using different imagery, we can look at the health of the stand. So we can see if there's fungal outbreaks or if insects are coming in. You can kind of pick out those patterns on a year to year basis, which is kind of cool, and we can track over long term. So as I mentioned before, restoration is a big part of this. So we're exploring different means of restoration. So historically, a lot of people that were trying to restore rice just bought mass amounts of seed from Wisconsin and Minnesota area where there's a lot more wild rice, and just threw it out. So they would buy it by the hundreds, thousands of pounds and just throw it out. And we call that a chuck and pray method. You buy it, you chuck it out, and you hope that it grows. But as I mentioned before, we're starting to see that seed from local areas might be doing better. If you can match some of those seed source areas to your restoration areas, you might have more luck. So we're trying to experiment with some of those things as we try to bring back rice to our area. We're also trying different things like transplants. So grabbing plants when they're in different stages from different populations and then transplanting them to new areas in hopes that if they're already pollinated, that they should be able to grow and drop their seed in these new areas naturally. And we had some good luck with that, but now we're dealing with some permitting issues on that we're trying to work through with Eagle, as well as some other additional restoration practices like invasive species control. I mentioned before that mute swans and carp and other things can have huge effects, and it's very obvious to see in this top picture here. That's a 6x6 steel cage, and you can see that that whole area was seeded there, and the only wild rice that's coming up is within that cage. So we're experimenting with different ways to do this to fence off areas and trying to identify what biota are actually having the impact and when they're having it, to be able to kind of better our tactic here if there's a way to increase our cage size, because obviously the cages are working, but we need a little bit bigger area caged off to be able to be successful. Other species that kind of compete with wild rice, we're looking at the proper ways to combat those. So whether that be Phragmites and spraying it, or narrow-leaf cattail and cutting it at certain times of the year. Another thing we do is purple loosestrife. We release the beetles to do a biocontrol. And then we're also looking at ways to remove carp. So whether that be electrofishing and removing them, or baiting them and trapping them. There's a couple different options there. So that's kind of a general overview of what we're doing here at the tribe. We were out harvesting all week, so now is the time that the rice is ready. It kind of differs between the state, but as you can see here, there's some pictures. At the bottom right is canoe full of harvested rice with a knocker. That's how we harvest the rice. You kind of lean it over the canoe and knock it, and it falls in when it's ripe. In the middle and the bottom there is our Monomin camp from a couple of years ago. Unfortunately, this year we weren't able to do that with the COVID situation, but annually we do a wild rice camp over on the other side of the state. So hopefully next year we can return to that. And then you can see some of our staff there doing some different monitoring that we do throughout the year.
Thank you, Alex. I don't see any questions in the chat box, Feel free, everyone, when you do have questions, to just put them in the chat box. But since I don't see any questions right now, we're just going to keep going. Let me share my screen for just a second. So our next presenter is Corey Lucas. He's the Institute's Stewardship Coordinator. But I do want to do one quick acknowledgement before we hand it over to Corey. It's just a land acknowledgement. And this is just something that we want to start doing, and just let people know that truth and acknowledgement are critical to building mutual respect and connection across all barriers of heritage and difference. We began this effort to acknowledge what has been buried by honoring the truth. So Pierce Cedar Creek Institute and much of Barry County now occupy the ancestral lands of the Pottawatomi people. And we want to respectfully acknowledge the Pottawatomi people who have stewarded this land throughout generations and continue to do so. So please take a moment just to consider the many legacies of displacement, migration, and settlement that bring us together here today. And we're doing a lot of this work at the Institute because of Alex and the Gun Lake tribe. And so we are very grateful to the tribe for bringing us in on these types of projects and letting us be a part of it. All right, so I'm going to stop sharing that. And Corey, I'm going to just start sharing your video if that works for you. Corey put together a great video about a little bit about what we do here at the Institute. So, all right, here we go.
Hi, I'm Corey Lucas, Stewardship Manager here at Pierce Cedar Creek Institute. We're standing out here today at our own lake, where's one of our sites for wild rice restoration. And so we came out here today so you can kind of get a look at what some of the habitat types might be for wild rice. In this scenario here, we have wild rice. It's actually growing on the north and south shore of the lake here. This is a man-made lake. It doesn't get deeper than about 15 feet deep. And what you're seeing here is a shallow portion of the lake where the lily pads end. It's about 4 feet deep. So we planted this wild rice in relatively shallow water, and that's really what you want to look for when you're trying to do wild rice restoration, is trying to maintain less than 3 foot deep water. So in this case, we planted it primarily in that 18 to 24 inches deep of water. It did real well. This is the first year we've seeded out here, and if you can see from the video, there's a pretty good patch of wild rice here, a nice rice bed starting here. And then over on the other side of the lake, on the north side of the lake, you can see it, the rice bed over there as well. So the reason we thought this lake would be a good lake to grow wild rice is because it's protected. So that's probably the number one or the most important thing when it comes to wild rice restoration, is finding a protected water body. It's really susceptible to boat traffic, to pesticide use, to animal herbivory. So this lake is kind of a prime example of a good habitat because we own the entire lake, so it's protected. We're not using herbicides on this lake, and it tends to not get huge waterfowl numbers. So we don't get huge numbers of geese or swans. They're here just in relatively low abundance. So we felt like those were good conditions. It also has some water movement through it. So there's an inlet on the west side, and we're standing here near the outlet. So there is some slight water movement through this lake. And that's one of the other considerations when you're talking about wild rice restoration, is trying to get a water body that doesn't necessarily have stagnant water, that it's got some either wave action or some just outflow. So those are some of the reasons we chose our own lake as one of wild rice restoration sites. Second week in September now, and right now is about the perfect time to harvest wild rice. Take a big stalk like this, they take a stick, and one stick you bend the rice down over, if you want to hold that. So you're coming through with your canoe, and they take one stick and they bend the rice down, and then they don't hit the rice, you hit the stick, and it shakes that seed head like that, and then that will drop the rice that's ready to be harvested. And the rice that's not ready to be harvested will stay on the stalk. And it's both protecting the plant and the seed, so as opposed to running your hand down that. And so it'll tend to look like this. You have the seed heads are starting to lose their shape. That seed's starting to fall into the water now. It's gone through what we call kind of its phases, right? So the first phase in wild rice growth is the floating leaf stage. So it'll grow up and it'll have these floating leaves that just float on the water. The next stage is elongation, right? So it goes from that floating leaf to extending a stalk up and shooting out of the water like you see here. And that final phase is the flowering phase. And we're just losing our seed heads now, so we've gone through that entire growth process for wild rice. What that rice will do now is fall off the plant, settle down into the substrate over winter. So it's stratifying during the winter time. It's going through that cold, wet process to help that seed germinate in the spring. So that's kind of the quick life cycle of wild rice. So we're out here in a different habitat today. So we talked a little bit about wild rice on our own lake and kind of the lake habitat for wild rice. We're out here on Cedar Creek where we have a slightly different habitat. This one's going to have more flowing water. It's going to be shallower in a lot of those areas. It's going to have a lot more organic material in the substrate. So those are kind of the main differences. We found wild rice initially here. Actually, Alex from the Gun Lake tribe found a species of wild rice out here 2 years ago, and that's kind of what kick-started this wild rice work for us. So we knew it already grew naturally in this environment. It was already here. So this past year, we spent time seeding some different areas along the creek with a couple different species of rice. So the palustre species and the aquatica species. We seeded them both in here to see how they did in this environment. What we found is palustres did actually grow in portions of the creek. It just was heavily predated by waterfowl. So we get large numbers of geese and swans and ducks back in these backwaters here, and they really focused in on that palustres, and they got to it before it got to that elongation phase. So that was our primary issue. It wasn't germination. We had plenty of germination. It was basically just animal predation. So we've expanded the seeding operations. We've done some seeding back on Massasauga Creek, which feeds into Cedar Creek, and we've had moderate success with planting aquatica back there. We can get it to germinate and get to seed stage, but just not in very large numbers. So we're going to continue to look at what specific genotypes might do better back there. But it tends to be away from waterfowl predation. So that's the bonus of having some of these different areas to experiment with. So this is just another example of where wild rice grows, and we're going to continue our work out here through the next 5, 6, or 20 years, who knows. So we're standing here in front of our wild rice propagation tanks, and many of you have seen these green tanks as you drive down the entrance to the Institute. These tanks were gifted to us by the Gun Lake tribe, and we use these to grow wild rice from seed. So what we typically do is in the third or fourth week in March, we'll come in here and plant wild rice in separate individual propagation bags with composted cow manure in there. And what we're looking at is there a benefit to growing wild rice and transplanting it into the lakes as opposed to just directly seeding into lakes? So we go through the same process here as what I talked about in the lakes. We'll watch it go through floating leaf stage. It'll get to elongation stage, and right when it gets to flowering, that's the moment where we like to transplant it. This year with COVID-19 restricting us in a few ways, we were only able to transplant that here into our retention ponds. And so we'll use those retention ponds as example or research sites for next year. This is the second year that we've done this propagation, and we've had high success with growing wild rice. And so we're optimistic about utilizing this method to restore wild rice on the landscape. When we were at the wild rice tanks, we transplanted the wild rice that we grew in those tanks to these retention ponds this year. We just did that work a couple weeks ago. And so what we'll do is we'll watch those plants. We've recorded data on how well they produce seed, how well they produce their flower head. And next spring we'll come out and look at whether those plants were able to reseed in these retention ponds. And it'll also be a great experience for us to learn if retention ponds are a possible habitat for wild rice restoration. So this is another one of those sites that we're watching closely and hoping to learn more about. There's lots of lakes like this in Berry County. A lot of people have these little lakes on their property that are protected. Some of them are man-made or manipulated by man. And I think there's a lot of opportunities there for private land work in Berry County.
Okay, so I'm going to stop sharing my screen. And Corey, if you want to turn on your video and also start sharing your screen, that should have given you guys a little bit of an overview.
All right. Are we good? You can hear me and see my screen here?
We're good, Corey.
So yeah, thanks for joining us today. There was a little video to give you an overview of what we do here at the Institute, and everything's been done through that partnership with the Gun Lake Tribe. So throughout my presentation, I'm going to stress utilizing partners that know what they're doing. Because we always had this idea that our property could hold wild rice as the right habitat, but we never knew it existed for 18 years till we got someone who knew what they were looking at out here when Alex came and visited. So that's just an important note there. So why do we care about wild rice restoration? Different people are going to have different goals. You talk to someone from the tribe and they're going to really, have that cultural importance, whereas you may get a duck hunter that, is just interested in providing waterfowl habitat. And we kind of do it all. We're interested in all of it. But it really does start with local cultural importance. Like Alex mentioned, this is why the tribe's here. This is why the Gun Lake Tribe is here, is because there was abundance of rice. So we want to try and bring that back. We want to increase the local abundance for those local tribal units. Alex talks about going up north to rice camp to harvest rice. Sure would be nice to have enough rice right here in their own neighborhood. So that's important to us. Also, waterfowl habitat. It's often considered to be the highest quality waterfowl food. It provides, habitat and food throughout the year. So Alex talked about, geese and swans are feeding on that plant and ducks at that floating leaf stage, but they're also feeding on the seed. So they're kind of benefiting from it in the spring and early summer, but also benefiting when they got to bulk up for migration. So very important waterfowl food. It provides excellent brood rearing habitat for ducks. When they go through molt, they're able to hide out in that wild rice and avoid predation. So it's excellent for that. And like Alex mentioned too, it's good brood rearing habitat for fish. And then also great ambush predator habitat. So you're thinking about largemouth bass, northern pike, they can hide out in that wild rice and then ambush their prey. And then there's something that's kind of new that I don't know that a whole lot of research has been done on it. With a decline in pollinators, we're really interested in what kind of habitat does this provide for pollinators. There's been some study in China on these large rice beds that shows a large diverse group of insects will use rice flowers. So that's something that we're kind of like, that's some new research we might want to consider. So how did we get started? I mentioned a large part of this goes to the Gun Lake tribe, a large credit goes to them. I contacted Alex back in August of 2018 and said, hey, we'd like to do wild rice work, but we want you to come out and look at the habitat. So he came out and actually found the state threatened Zizania aquatica in Cedar Creek. So that was kind of a cool moment for us. And that started this partnership. So in working with them, we kind of developed this propagation and transplanting program that next spring. And we were able to successfully grow wild rice in these propagation tanks. We kind of experimented with different substrate and different seeding rates to see if there was an issue with lack of nutrients or with competition. And so through that, we were able to narrow down how we wanted to propagate rice for 2020. Again, we successfully grew the wild rice, but due to some of the COVID restrictions, and then like Alex mentioned, there's some permitting that we got to get figured out. This is all new to everybody, pretty much just transplanting wild rice. So figuring out that permitting process is a hurdle we're going to get over this year. And we used one substrate and we used higher seeding rates based on what we found in 2019. So that was kind of our March 2020 transplant or propagation program. And then we also began seeding this year in our lake. So we hadn't seeded prior to March of 2020. And I'll share kind of our successes and failures there. So in our seeding program, we had seed sourced from the Gun Lake tribe through their harvest. We planted that, we broadcast seeded that in Brewster, our own lakes, and Cedar Creek. So Palustris was seeded in all three of those areas, and then just Aquatica was seeded in Cedar Creek. All the sites germinated seed, which was promising, and we got really excited in the spring. And it was even out competing with, you know, the vegetation, which was kind of surprising to us. So spatterdock and lilies, it was growing right up through that and doing fairly well. So it was promising early on in 2020. June, July looked pretty good. Things changed a bit on Cedar Creek. You can see from the picture here. We had it get to right about the beginning parts of that elongation phase, so shooting out of the water. Some of it still in floating leaf, and it just got hammered by geese. And we have some trumpeter swans. So, you know, you're thinking about, well, you're growing this stuff to attract the waterfowl, and then you're like, you know, shame on you for eating the wild rice. So I think we want to get it to a point, we want to baby it to a point where it's large enough to be able to handle this herbivory from the waterfowl, and we're just not there yet. So our plan is kind of to reseed these areas in 2021 and then put exclosures or enclosures around it, like Alex showed some of his for next year, just to get those rice beds started. There's a close-up video or a close-up photo. You can really see how this is waterfowl, right? Because it's above the water. You know, it's not muskrats. It's quite a ways out of the water, so it's most likely geese or swans. So our own lake, and this is our success story. This was after our first season, or during our first season of seeding. We seeded approximately 10 pounds of rice on only a quarter acre, and it literally grew everywhere. I mean, we could brush our hands off of rice, it would grow right there. So a really, really good habitat for it. And again, we're kind of hypothesizing that it's because we had a protected water body. There's no pesticide use around that lake. It's got all natural shoreline. There's no boat traffic, so there's no motors ripping through there. There's very few invasive species. You can see some spatterdock and lilies in the picture there, but very low abundance of purple loosestrife. There's no phragmites, so some of that, no milfoil. So very few invasives, relatively low waterfowl use, so this doesn't get heavily used till later in fall migration. Some swans and some geese, but very low numbers. And then, you know, like the researchers we'll talk about later, we're kind of narrowing in on these genotypes and figuring out that, well, maybe there's some genotypes that just flat out do better in restorations. And we're trying to use those and experiment with those genotypes that we think are better suited for our climate and our habitats here in this part of the state. So in 2021, we'll plan on reseeding and monitoring this existing rice bed, but really, really good results for the first year. There's kind of another picture of it. It's also on the other side of the lake. You can see it kind of off in the distance there. Made it all the way through, the seeding stage, and we were just out there this week, and it's dropping its seed. So I wanted to just put together a couple slides here that if you were a landowner or you were a land manager and you're like, I need a checklist. I just want to go through a checklist and kind of see if I've got the right conditions to grow wild rice. And so this is, and there's probably more to it, but this is kind of a simplistic approach to assessing the habitat. Does it stay wet for more than 120 days during the growing season? That's essential. You don't want it drying out. It doesn't need to be flooded necessarily, but the soil definitely needs to stay saturated. It needs to be in less than 3 feet deep water, preferably shallower. We're seeing it grow better in that like 18 inches. Protected from pesticides. So, if you've got a bunch of lawns sloping down to the lake, or you've got a lake that's doing weed treatments, it's going to be a uphill battle. No or limited boat activity, specifically motorboats. They're really, let's say, gently rooted. They can be uprooted very easily. That substrate that they grow in isn't like growing in upland. It's not a fixed soil. It's kind of saturated and easy for that rice to get uprooted. And then limited or no competition from invasive species, and then organic substrate. So having a good layer of organics there for it to grow in. That's kind of the habitat aspect. And then, how do you go from there? You've identified that you've got pretty good habitat, but you got to take the next step. So I would suggest connecting with a partner organization, at least like share some ideas, possibly get, depending on the habitat, possibly get some help, sourcing seed. And so there's the two that you've got in front of you today. So feel free to reach out to us. Get a good seed source and keep it good. And that means, the researchers will talk a little bit about genetics, and Alex touched on it too, is that we're finding that maybe there's certain genotypes that work better. So, get it from a good source. And, buying seed from the cheapest supplier, maybe that's Minnesota, may not always be the right idea. We're still learning more about that. Remember, seeds must be stratified, so they got to stay wet through their life cycle, and they have to go through that cold period for germination. So don't keep them in a bucket and keep it in your garage. You need to have it out. We actually put it out in the water over winter, where it's got some flow through it. So that's important too, is just to keep that seed wet, and then stay connected. So, Alex said that a lot of this we're just learning, and we're actively researching this. All of our partners are, and so stay up to date. Get tied into these programs. This is a great way to learn more and get those contacts, and then contribute to research if you can. If you're looking to do wild rice and you've got a good habitat and it's protected and it's a large enough water body, think about possibly being a research site or a restoration site, and or a harvest location. So that's kind of my checklist for you landowners or land managers wanting to do wild rice restoration. And there's some of my favorite waterfowl pintails there on a rice bed down in Texas. So waterfowl definitely like to use it during fall migration.
Awesome. Thank you, Corey. I know we do have a couple of questions, but we are going to leave those to the end because we're running a little shorter on time. got a lot we want to get through today. So our next speaker is Dr. Brad Swanson from Central Michigan University. Dr. Swanson, if you want to unmute yourself, and you are totally on top of things, awesome, and start sharing your screen, you should be able to just start sharing.
Okay, hopefully everyone can see that then.
You are good.
Okay, brilliant. So basically what my lab has been working on wild rice for about 10 years now. We've worked with Little Traverse Bay bands, Little River bands, Pokagon bands of Pottawatomi, Gun Lake, a bunch of different ones. We've worked on ecological aspects and a lot of genetic aspects as well. And we're going to talk a little bit here about the genetic stuff that we've done. I will say just this last year, I had 2 students working at Cedar Creek and in other places around Michigan, working a lot with Alex and the Gun Lake tribe, looking at ecological factors. So we were trying to compare regions of lake that had rice, regions that didn't have rice, seeing if there was any ecological, so basically soil conditions or water conditions that might impact why rice was growing in some areas and not others. We haven't finished all of our analyses yet there, so if you want to learn more about that, I suggest you come in and watch those presentations on the research that Ellen talked about earlier. So if we think about rice, okay, now it doesn't want to move either. It's just, yeah, nothing works. There we go. So Linnaeus, the father of taxonomy, described 2 species of North American rice, Z. aquatica and plustris, in the 1750s. Then in 1908, Hitchcock accepted the Linnaean names, but in what caused a whole lot of confusion, he actually flipped their orders. So what Linnaeus had described as aquatica, he called plustris. What Linnaeus called plustris, he called aquatica. And this has caused some problems ever since. Okay, so then in the 20s and 60s, a couple of other individuals, they collapsed both species of rice into a single. I'm trying to get this so I look like I'm looking at you folks at least. Collapsed everything back into a single species with varieties. And then in the 60s, Doyle converted them again back into aquatica and plustris. He used the original name suggested by Linnaeus, so altering again what Hitchcock had said. So now we have flipped the names of these species 3 different times, and then had a series of subspecies associated with them. Where we're at right now is Terrell's 1997 version that says aquatica and plustris, and these still use the names that Linnaeus came up with. When you look at the distribution of them, they overlap pretty substantially in parts of their range. And this can cause problems also for why people can be confused about what type of rice they really have. We have this unique little population down here in Texas. That would have been the one that Corey just showed. And that is Texanum. So it's a unique species, Texana, of wild rice. When they do grow together, we do see them as hybridizing, and they do tend to produce fertile offspring. So depending upon how we really want to think about this, think about it from the standpoint of what a species is, there's several people who would suggest then that these are not distinct species because they can exchange genetic material between each other and produce fertile offspring. So when we look at why Corey and Alex were talking about some of the uniqueness that we might see with the genotypes for rice in Michigan, one of the issues is when you look at Michigan, we are an isolated peninsula on 3 out of our 4 sides. To our west, we have Lake Michigan. To the north, depending on where you are, it's either Lake Superior or Lake Michigan and Huron. On our east, we have Lake Huron and Lake Erie and Lake St. Clair. But the idea is that Michigan is really restricted from gene flow coming in most directions. So this suggests that we might have evolved some unique strains of rice that we don't see anywhere else in the world. And that was one of our main reasons for starting some of this genetic work. Now, we also have population declines throughout the rice, as Alex was talking about. and this can cause and be especially problematic for rice populations, where they can enter what we call an extinction vortex. And that happens when we have either habitat fragmentation or habitat loss. That means we end up with smaller, more isolated populations. So there's less movement amongst our populations of rice. As they're smaller and isolated, there's less dispersal or less movement of individuals between populations. When we reduce that movement, we start to see inbreeding, basically mating with closer relatives than you would perhaps want to, and that tends to decrease genetic diversity. As we decrease our genetic diversity, we end up reducing survival and reproduction, and in the long term, we lose adaptability to changing conditions. And with global climate change, we know that that adaptability is going to be a major component of the survival of wild rice. As we see reduced survival and reproduction, though, we get our smaller population size again. That smaller population size then just keeps feeding into this cycle, which can eventually end in localized extirpation or even possibly extinction of the species. Now, as people have mentioned, and we saw with the taxonomic debate, there's questions as to how many species we might actually have. And this is one of the questions our lab has been interested in, is how many species do we have? How isolated are each of the populations in Michigan? And what might we predict for the future? Now, most people are not going to know what this necessarily represents, but this is essentially it's kind of a genetic map of looking at different genotypes. So genotype is just the genetic makeup of an organism. And what it's trying to show here, these are in birds, but what we want to look at is the differences that we see between them. Each one of these circles that we see here represents a sampling location, and the white circles are black-backed orioles, the grays are Baltimore orioles. So each one of these circles represents a unique genotype. And what you can see is all of the white ones are connected together and all of the gray ones are connected together. When we see a situation like this, this says we have good, solid species. All of the genotypes associated with the white circles are always black-backed orioles. All of the genotypes represented by the gray circles are always Baltimore orioles. We don't ever see a black-backed oriole with a gray circle, and we don't ever see a Baltimore oriole with a white circle. And then when you look over here, we're looking at other aquatic plants, and you can see the same type of thing, where we have representations of two different species, and they're always well defined, so that the black circles are always one species, the white circles are always another species, and we never have any mixing. So this is what we were hoping to find when we looked at the rice in Michigan, the wild rice. But we weren't necessarily expecting that, because there had been previous work done in 2015 that looked at rice across a large chunk of North America, well, of the US. And what they ended up finding was, this is the latifolia, I'm sorry, another unique species. You see it separates out all by itself, definitely a good species. Texana here separates out all by itself. It's a good species. But then the problem we have is we see mixes of, sometimes we have Z palustris attached to Z aquatica, going back to Z palustris. Instead of all of these Z palustris clustering together, we see a mixture of the aquatica and palustris. So this suggests that there is not a good separation of these two species, and that they might need to be collapsed again. Now, the one thing that was interesting was zoo never had any samples from Michigan. So this was what got us going to try and see what was going to happen. As we looked at Michigan wild rice, we wanted to see where we fit into this genetic makeup that we saw here. And we also wanted to see if we had anything unique in Michigan. So I had a graduate student, Whitney Belaski, who sampled 12 to 20 some populations of aquatica and palustris. And one of the very cool things that she found was she did find unique genetic haplotypes, so unique genetic signatures that are found only in Michigan. And that's represented by these letters that are in bold: AB, AC, AA, AD, and AE. And we found one unique genetic type for Aquatica and 4 unique ones in Palustris that weren't found in any of Zoo's work that really sampled almost all the rest of the US. Now, the other thing that Whitney found was she started to find other samples that were representing possible hybrids. So she was finding haplotypes, the same DNA sequence in both Aquatica and Palustris. So for example, this haplotype D here, we see it's found in both Palustris, the white, and Aquatica, the gray. So this, and same with sample P. So this suggests then that there's exchange occurring between these two haplotypes, between these two species. When you look at the distribution, and one other thing, I have a current grad student, Dan Carlson, who's working on this as well. And one of the cool things is he has found another unique genetic strain of Z. palustris up in the Pigeon River. And he's added probably another 100 samples total to the number of rice plants we've looked at. And the cool thing is the new sample for Z. palustris A found is only being found up in Pigeon River. So we might have another unique Michigan haplotype. And so this then is suggesting that Michigan is being isolated by the Great Lakes, and we do have some unique genetic strains of rice in Michigan. And going back to what Alex and Corey was saying, if we have unique genetic strains, it suggests that we might have some unique adaptations that are occurring in Michigan. And that might be why when they're planting rice, they plant rice that's growing in Michigan, as opposed to buying from another tribe to plant, that it comes out looking better. All we want to do here, all I'm trying to do here is show you some of the unique samples that we have. And you'll have to excuse, the circles aren't quite exactly where they belong because of how big they are to make them visible. They're all slightly offset a little bit. For example, A is not actually on Beaver Island or any of the Manitou Islands. It's actually up here. But what it shows you is you can see the different types of haplotypes or the different haplotypes that exist across the US and even into a bit of Canada. The light green haplotype is found throughout its entire distribution and is the most common haplotype we have in Michigan. But the really cool part is the unique haplotype that we found, haplotype AC, where we found it in 2 unique at high densities in 2 populations in Michigan. So that suggests that these populations are likely to represent true ancestral populations of Michigan's wild rice with respect to Aquatica. It doesn't mean that any of the areas where we have the green are not natural and old populations either. It just means that they aren't unique relative to what we end up seeing here. And that's looking at Z. palustris. We can kind of see the same thing. This time though, we have our 4 unique haplotypes and the one that Dan, my student, just found way up here in Pigeon River again. And you can see that some of them, the dark blue is relatively rare. The gray one is relatively rare, but the yellow haplotype is relatively common in some of our populations. And then our last one, the AE, is relatively rare as well. So the thing that we've found is that Michigan does have absolutely unique genetic strains of wild rice relative to other populations, but they tend to be in relatively low densities at any of the sites we found. So the fear that we have is that we're going to start losing these unique genetic strains before we actually recognize them. And that's one of the things that the tribes have been fabulous with, is trying to make sure that we are harvesting from areas where we have unique genetic strains so that we can actually bring those back into the populations. And with that, I am done. And I'll stop sharing my horrible screen situation.
Thank you, Brad. We appreciate you sharing your research with us today. As I mentioned, we do have one more speaker. Our last speaker is Charlene Partridge. She's part of Anna's Water Resource Institute at Grand Valley State University. Charlene, if you want to start unmute yourself and start sharing your screen. We will answer a few questions at the end. I know there's a couple of people that have questions in the Q&A box.
All right, so first I just want to say thank you so much for the invitation to speak with you. And I'm just going to talk a little bit about our work in collaboration with the Gun Lake Tribe, specifically Alex, looking at the genetic relationship of some of the populations that they're managing in Michigan. But before I start with that, basically what I want to do is just take a few minutes to talk about why looking at or understanding the population genetics of these populations you're trying to manage are actually important. Oops. Okay. So why is understanding population genetics important to conservation? And I just want to give you a few examples of how these types of scenarios have helped bring back populations and kind of what you can use this information for to help manage current populations. So first of all, understanding population genetics can aid in reintroduction assessment. So one of the classic examples of this is the reestablishment of the Blackfoot Ferret in the Western United States. So in the 1970s, the Blackfoot Ferret was thought to be extinct. And then in 1981, a remnant population of Blackfoot Ferret was actually found in Wyoming. And they took a couple of individuals from that population and they moved them to zoos to start an actual breeding program. And which was actually a good idea because a few years later, the remnant or the rest of that population succumbed to disease. So they took 18 individuals, started this breeding population, and what they did is they created a computerized matchmaking system where they took the genetic information from all 18 individuals and mated them so that they increased or optimized the amount of genetic diversity that could potentially be produced in the offspring of those population and the offspring of those individuals, and then released a genetically robust population that would be able to adapt to these wild environments. And so we saw success with this program initially where we saw the increase of, or the population numbers of these ferrets increase, reaching up to around 1,000 individuals. Unfortunately, we've started to see a decreasing trend in these populations now, but that's due to the spread of salvitic plague that's actually in these areas. One other example of how we can use genetics to kind of better understand how to manage populations is by looking at source populations for the potential for reintroduction. So that's kind of some of the stuff that we've been talking about. So can we identify populations that we know will do well when we move them to one area to the next? An example of this is the restoration project for reintroducing Atlantic salmon back into Lake Ontario. So Atlantic salmon became locally extinct from Lake Ontario in around the 1800s. And in 1990, the Ontario and Canadian government decided it wanted to reintroduce Atlantic salmon back into Lake Ontario for fisheries. So they started this in the 1990s, and over a few decades, they noticed that it wasn't successful. So our Atlantic salmon were not repopulating, they weren't successfully reproducing in Lake Ontario. So the question became why? What's going on with these populations, and why aren't our restoration efforts actually working? And there are two main problems with the way this happened. Number one, Lake Ontario of today is not the same as Lake Ontario of the 1800s. So there have been drastic environmental changes that have happened over that 100-year time span. Number two, when they were actually looking to reestablish, they were using hatchery fish to repopulate Lake Ontario. So there are a few things to know about hatchery fish, and these were multiple generation hatchery fish. So there's strong selective pressure on hatchery fish, which typically selects for traits that work well in a hatchery. So docile fish don't move around a lot. Well, those traits aren't that great in a natural population, right? So you want fish to be energetic and active and aggressive. And so what was happening is those traits that are very important for wild populations were being lost in the hatchery. And there wasn't enough genetic variation in the hatchery fish for those populations to evolve to the environmental changing conditions that we're seeing in Lake Ontario. So what they're doing now is, instead of just looking at one multi-generation hatchery population for reintroduction, they're starting to look at different populations, try to understand which of those populations contain the highest amount of genetic diversity and their traits of interest, and use those fish to restock Lake Ontario. And that's actually having more success in their efforts. And so those are just two examples of how you can use genetics in terms of managing and conserving populations. But those are kind of examples once the population has already started to crash, right? So how can we use these tools, these genetic and genomic tools, to kind of help manage current populations, so before it actually gets to that brink of loss? And there are a couple of different things that these data can do. They can help you identify how populations are connected, so they can tell you how closely related some populations are to other populations. They can tell you how much gene flow is happening between populations, or whether or not you have relatively highly fragmented populations with high levels of inbreeding. They can identify levels of adaptive potential. So this is kind of used, or genetic diversity is kind of used as a proxy of adaptive potential. If you have a population that's really genetically diverse, then theoretically that population should respond better to shifts in the environment. And this becomes very important in terms of climate change, as we know these populations will likely experience different environmental conditions over the next 50 to 100 years. And then, as has been mentioned a lot over the past hour, in some cases it can help identify species. So if you have two species that look very, very similar, and it's hard to taxonomically look or visually look and identify them, at times it can help you identify one species from another, or it can actually identify potential hybridization between two species. And so I'm going to briefly go over this since you've already seen it. So we've talked about the populations of wild rice in the US, and this is just a modern distribution map looking at the distribution of Aquatica in yellow, of Palustris in red, and then green is their overlapping region, and that's kind of what we're most interested in because Michigan is included in that area. And then this, we know, like Brad had talked about, we know a good amount about population genetics in Michigan. So this is just kind of a combination, and you've seen this map a couple of times, looking at the genetic variants of both Aquatica and Palustris throughout Michigan and throughout the rest of North America. And so, as Brad mentioned, you have some genetic variants. So these colors represent different genetic variants of these species. And so you can see some of those genetic variants are pretty broadly distributed. So the genetic variant of Aquatica A, you find it all throughout, the Midwest, up into Canada, and then all throughout the Northeast, on down to Florida. So you have some genetic variances that are everywhere, but then, and you see the same thing for Palustris. So the genetic variant, oh, I can't see it, sorry. My little window that shows everyone is kind of hiding this key. So the purple one, the purple genetic variant is broadly distributed in terms of Palustris. But then you do find these unique genetic variants. And for Aquatica, you have the AC haplotype, or the AC genetic variant that is unique to Michigan. And then you also have a couple AD and AE and AA of the Palustris that are unique to Michigan. So Alex and I started talking about a year ago, and they had some populations that they were interested in managing, and they wanted to know what they were. And I'm a molecular ecologist. I have a sequencer. I was like, sure, let's work together and see if we can figure out what these populations are. So Oops. So they were mainly interested in looking at for the populations that they're currently managing. And what we did is similar to the Shu et al. Study in 2015 and the Bolesky work in 2017, we sequenced the same 3 genes that they sequenced. And these are genes that are located in the chloroplast genome. So one thing to note is, so the chloroplast genome, typically all of your genetic information that kind of is the important information to build you is what's called nuclear DNA. So that's the DNA that's located in the nucleus. So when we think of genome, that's normally what we're referring to. But there are separate genomes in a cell as well. And so in animals, they have mitochondrial genomes. In plants, they have chloroplast and mitochondrial genomes. So the genes that we sequence to be able to look at the genetic relatedness among these populations are part of the chloroplast genome. And this is important because chloroplast genomes tend to be maternally inherited as opposed to your nuclear genome, which is both maternally and paternally inherited. And so what this means is when we're tracking this genetic relationship, we're tracking the relationship of the maternal line for this plant. And then we used those sequences and compared them to all of the sequence from the previously identified genetic groups. So basically we created a database of all of the sequences that are currently found on this common database called GenBank. So anything that was related to these 3 genes associated with Palustris or Aquatica, we downloaded, and then we compared our sequences to those sequences. And so we were tasked with 2 things for this study. Can we determine the species, not by phenotype? So in our lab, we didn't know the phenotype between these 2 populations. So can we just determine the species purely based upon the underlying genetic information? And then can we identify any of the Michigan-specific variants and whether or not they're present in those populations? Because like has been mentioned previously, those are the ones that we may be interested in preserving. And so here are our 4 populations that we looked at. And so what you can see here is that there wasn't a huge amount of variation in the genetic variants that we saw. So this population, everyone aligned to Aquatica with the genetic variant A. Some of them we couldn't actually distinguish between. In many of these, it's only 1 base pair difference that distinguishes between the haplotypes or between the genetic variants. So in the case of this population, it came closest to Aquatica or genetic variant H or the D variant. And again, the D variant can be for either Aquatica or Palustris. So that kind of makes it hard to identify whether or not it's an Aquatica species or a Palustris species because that haplotype is present in both of them. And then for this population, most of the individuals aligned to the A genetic variant, and then we had some that were either A or E. But the one thing to note is that all of these haplotypes or all of these genetic variants tend to be more broadly distributed, so they're not Michigan specific. And then we found a couple that were undetermined, so they were not present in our database, so suggesting that they may be unique haplotypes. And then when we look at our northern population, we do see some of those unique variants. So AE was present in roughly around, I don't know, around 19% of the population. And then also we had one group that's labeled the L haplotype. And from the data that we downloaded, we had genetic variants Ls that were assigned to both Palustris and Aquatica in the information that we downloaded. So again, whereas it looks like from the literature, when you look at it, it looks like that Palustris is, or it looks like that L genetic variant is more Palustris. When you download the data from GenBank, there appear to be some Aquatica L haplotypes as well. And then we had this unknown or undetermined genetic variant as well, which may be a unique haplotype, similar to kind of the stuff that Brad has seen as well. So we have some, or from the data, or from the populations that the Gun Lake tribe is managing, it seems like there are some of these unique Michigan haplotypes and some of haplotypes that we weren't actually able to assign, suggesting that they may be unique as well. But we also, for many of these, had trouble determining whether or not that sample that we got was one species or the other, because the haplotypes are present in both species, or the genetic variants are present in both of the species. So what's next? Well, I think it's important to remember that from the data that we're looking at, we're actually only getting half of the story. Like I said, the chloroplast regions that we're looking at, these are only inherited by one parent. So we're missing the other half of that story, which is the paternal line. And so what we want to do now is to kind of expand on this further and kind of identify who these populations are at a more like fine scale level. We want to actually use a combination of nuclear markers, which are biparentally inherited, and our chloroplast markers, which are maternally inherited, to kind of gain a better understanding of these populations. And so the questions we want to know are, how related are these populations to one another? Is there any gene flow happening among these populations? Or if they're severely fragmented, are they also severely inbred, and kind of how do we manage that? One important thing is how much genetic diversity is present in these populations. So if you do want to restore a population, you want to restore robust populations. So you want populations with high levels of genetic diversity that can buffer from environmental change and adapt and evolve to be able to meet those environmental changes that we know are going to happen. So are the populations that are working well more, are they showing higher levels of genetic diversity, or is it a genotype specific component? And so that's something that using this combination of nuclear and molecular and chloroplast markers will allow us to start to ask and address. And then including both nuclear and molecular marker, or nuclear and chloroplast markers, to really look and see how distinct these two species are at the genetic level. So if the phenotypes are becoming more intermediate between these two species, is that because of environmental variation driving differences in those phenotypes? Because that's one way it could happen. Alternatively, we could be getting a lot of hybridization. So a lot of these populations where we're seeing these distinct overlapping genetic variances may be hybrids of the two species. And how you manage distinct species versus how you manage hybrids may be very important, because depends upon how healthy those original parent populations are relative to the hybrid populations. So how you may actually manage those, or so those populations may require different management techniques or methods. All right, so that's kind of all I have for you today. I would like to thank Alex for bringing me into this project. It's been really fun. It's a lot more complicated than I initially thought. I was like, sure, we can use genetics to tell you exactly what species it was. And then it kind of became very confusing. And then I'd also like to thank my lab technician, Sindel Parks, for doing a lot of the lab work for me.
Right. Thank you, Charlene. I think if you stop sharing your screen for a second, we'll be able to see everyone, all of our panelists. And we have 3 questions in the Q&A. We'll go through these questions. All right, our first question that we had was about genetics. So Charlene, this is for you. Are the 2 areas of undetermined types found in the southern versus northern the same, or are these 2 separate types?
They are 2 separate types. Yeah, so they're very similar to the genetic variances that are there. Like they're one base pair different, but we just can't say that they're one versus the other because they have to match perfectly for us to assign it. That's why we have a few that are like D or H, because it matches perfectly for those except for one place. And so, yeah.
Awesome. All right, our next question is for Alex. If you could discuss what you have tried for cattail removal and its success in terms of for the wild rice.
We're kind of just getting into this. We've tried a couple things. Obviously, glyphosate is the best, but we're trying not to use that when possible, just because there's the leftover chemical in the soil can inhibit rice growth. We did try cutting below the water line in the fall. That kind of depends on the size of the stand. We have one stand that's about 2 acres. So to try to cut all that in 1 year is a lot of man hours. And then the other issue we run into is if there's part of the stand that's above the water line, it's not really effective. So it kind of depends on the situation you're in, I guess. If it's a small stand on the edge of a lake, it's pretty easy to go through and cut it below the water line and drown it out, and that's pretty effective. But if you're dealing with a stand that's in a water that's kind of variable and it goes out of the water line, you almost have to spray it from what we found.
Thanks, Alex. The next question is actually also for you, Alex. And this will be our final question for today. Does the Gun Lake tribe actually harvest the rice for consumption, or is this just a project to see if rice could be put back into its natural environment?
Yeah, we do. There's obviously a lot of our citizens go out on their own and harvest rice throughout the state. The locations of a lot of these stands are held pretty tight for obvious reasons. But we harvest rice at about 90% of the rice that our department harvests goes to restoration. A little bit we keep for ceremonies and different things. And then our language and culture department here at the tribe also goes out and harvests, depending on the year, how much, but usually around 100 pounds. And they use that for different gifts and ceremonies and whatnot to give to some of the tribal elders and other citizens. And kind of one of the other things I'd like to mention here is that a lot of it, you can see through these maps, the tribal territories for different tribes kind of vary throughout the state. And some of the better beds are up north in different tribes' territories. So one of the things that we do is we create a bundle and we give that to those other tribes, and that kind of, they give us the permission to go harvest in those areas. So some of that rice goes to that as well.
Wonderful. I know we've covered a lot of ground today. I want to thank all of our panelists, our speakers today. I know Brad had to leave for another engagement because we were running a little late. So thank you guys so much for sharing your knowledge. I want to thank everyone on the call for coming in today and learning about wild rice. So thank you everyone so much for helping to learn how to bring back the wild rice.